get up here. Well, thank you very much. It is indeed a privilege to be here and also to bring a real hero. I like to think of patients as astronauts in the sense that they've been going forward and trying some challenging things that we come up with in the lab, and if nobody let us get into the clinic, we wouldn't be anywhere. So that bridge has to have somebody that's willing to walk across it. And that's what our patients have managed to do. I do clinic once a week, sometimes a little bit more than that, but I spend a lot of time in the lab. And each time I'm in clinic, I'm reminded of how important what you've done is, Bob. That as the architect of Prop 71, with very forward-thinking philanthropists who care about humanity, we can make this a lot better. So it's not just about ending human suffering, it's about giving people a different idea about what their reality should be. I don't believe in the new normal for cancer. I think people should be healthy. I think the best palliation is cure. And I'm reminded of that, just having come off of 2 weeks of inpatient service, which was a little bit horrific, I've got to say. And because of what you've managed to do, Bob, and others in the room who haven't given up, who think there is a better way, I'd like to tell you about some of the things we've managed to accomplish. So we've done 7 clinical trials that have really emerged as a result of CIRM funding, philanthropy, and some very forward-thinking people in industry who I see in the front row here. So we're very grateful for their opportunities. So with great opportunity comes great responsibility. And I'm reminded of that when we get into the clinic, but also of the healthcare economics of cancer stem cells. So cancer stem cells basically were first thought of in 1994 by John Dick, our collaborator on a major CIRM grant, where he suggested that it was only a minority population within the cancer that could really propagate disease. And the problem with these cells is they could clone themselves, they could become self-sufficient because they were dormant in protective niches where they learned to survive. So although we have therapies, small molecules that people can take chronically, patients get resistant to these therapies, unfortunately, with greater frequency than I'd like to admit, because of the cost. So if you look at a disease called chronic myeloid leukemia, where people can take a single pill called Gleevec, the problem is it's $100,000 per year. And the cost to the individual patient can be as much as $2,000 per month as a copay. That's a problem. Anyway, so the cost is exorbitant, and I think, unfortunately, they're not effective because people can develop resistance mechanisms. So these bad seeds that lead to resistance are really what we're targeting in cancer, not just in leukemias where it tends to be easier because we can sample the disease from the blood and bone marrow, but in solid tumors. And that's really where I think we need to go. And fortunately, we have very thoughtful philanthropists like Denny Sanford and Malin Burnham at UC San Diego, where we're given great opportunities because they can see we're not there yet. We still have to do more clinically to get our ideas into clinical trials and really test them rigorously. Now, what we've been working on is targeting these leukemia stem cells in the niche, and there are a number of pathways that we find are activated. And I'll go through some brief vignettes about these, but basically we've discovered that embryonic pathways tend to be upregulated in leukemia stem cells that allow them to become dormant, to clone themselves, or in other words, self-renew, and then survive. So the first story, and Teresa will help me with this story after I finish showing the slides, is about a JAK2 inhibitor that was developed in San Diego by a local company called Targeten, and we came in when they were trying to decide what they should use it for. We suggested that they use it for JAK2-driven, what are called myeloproliferative neoplasms, which just means you make too many of one type of blood cell in the bone marrow. And that may sound innocuous, but I can tell you it isn't. People scar down their bone marrow, and then they can transform to acute leukemia. So with the CIRM training grant, we were able to have fellows that did this work, and then we got the first seed grant targeting cancer stem cells, and we're able to take this work where we could overexpress the JAK2 mutation and show that this targeted JAK2 inhibitor reduced engraftment and reduced the mutation. That led to 5 clinical trials. And one of them is the one Teresa's on, and she'll talk about this at the end. Basically, what we saw was a decrease in the JAK2 mutation, a decrease in the high white count that tends to be the sine qua non of the disease as it progresses to scarring of the bone marrow. And we finished phase 3 trials. Now, the reason we were able to do this in San Diego with such alacrity is because we're surrounded by biotech companies. We're one of the biggest hubs in the US for biotech. And so what happened with Targeten, and this is something that Bob was alluding to, people notice when things start to work. big companies noticed. So there was a $560 million acquisition of Targogen by Sanofi. So now that's investing in the state. And I guess, Drew, they met their milestone, so you might get a payment, or you already did. Anyway, this led to another very important project for us in terms of thinking, how do we come up with combination strategies to eradicate these leukemia stem cells forever, just get rid of them. So this was a grant between Canada and California that we feel very fortunate to share with John Dick. It was really aimed at getting rid of these cells that tend to self-renew in the bone marrow niche. Now, Irv and I discovered in 2004 that leukemia stem cells in a disease called chronic myeloid leukemia tend to activate beta-catenin, which is a self-renewal gene that should only be active in our normal stem cells. However, in the context of this leukemia, it gets activated at the wrong time in the wrong place. So he and I asked, why is this happening? And found that there was a problem with splicing of a gene that normally shuts beta-catenin off. Now, the reason SERM was so important for this is we couldn't discover this in mice. We splice more than mice. So this was a transcript we didn't see in mice. We only found in human leukemia stem cells. And so we wondered, why is this so human-specific, this missplicing event? So we started digging again through this collaboration with Canada, where we work with Tom Hudson at the Ontario Institute for Cancer Research, and also Mark O'Mara in Vancouver, and found that there was upregulation of a gene that edits our RNA. So when you think mice are supposed to live 2 years and humans are supposed to live more than 80 years, it means that we have to regulate our gene expression more like a thermostat than an on-off switch. And this editing gene called ADAR1 edits our RNA, so it keeps our RNA intact and prevents us from making mistakes with that. The thing is, when it's upregulated, it makes cells clone themselves. And so what we found earlier this year was it led to malignant reprogramming of these cells in CML. And if we knock it out, we drop the capacity of the leukemia stem cells to clone themselves. And that's what Fei Jiang showed in the lab. So they have lost that capacity. So the interesting thing about this gene, ADAR1, is it's actually activated, at least in part, by JAK-STAT signaling. So what we found was if we use the same JAK2 inhibitor I mentioned earlier together with a BCR-ABL inhibitor, we can actually reduce the capacity of the leukemia stem cells to survive and propagate themselves. So this is a sort of repurposing. And so some of the drugs that we discover in one context also work in other contexts, depending on the combination that you use them with. Another collaboration that stemmed from SERM funding was the work on the sonic hedgehog pathway. So this is a pathway that we have upregulated during embryogenesis, and then it should be shut off. And what really happens here is it doesn't get activated appropriately, it gets activated inappropriately. So Tanish Derea showed in mouse models of chronic myeloid leukemia that this pathway was activated, and then we showed in human cells that it leads to an increase in GLI2, and then what our collaborator John Dick in Toronto showed was that GLI2 not only allows us to detect the leukemia stem cells, it allows us to say, hey, they're actually responding to treatment. So this led to a phase 1 clinical trial. And what we saw in the phase 1 clinical trial that I'm the senior PI for was that the people that had a higher GLI2 expression actually had a lower survival. They had a worse prognosis. And you can see from the patients we accrued, we had an average age of 69. These are hopeful, fit people. They want a pill for their leukemia. They don't want to be patronized and told they have to lose their hair and have a bone marrow transplant as the only option. So you can see that the results were dramatic in acute myeloid leukemia, where we had a 50% reduction in leukemic blasts in 7 of 28 patients, which is actually very good for that kind of therapy. So again, this has been funded by quite a number of SERM grants. And I just wanted to show you part of this video because it shows you a patient that has actually gone through this.
Today, this grandmother is thriving. The chance meeting in San Diego is saving lives.
Live from the Scripps Studios, this San Diego's news source, 10 News.
And doctors say that she had about 2 months to live, but that was more than 2 years ago. A local woman now is alive and doing well. New at 6 o'clock, 10 News reporter Michael Chen looks at the local partnership that she credits with saving her life.
This is a symptom.
This bruising is one reminder of a disease that turned Linda Lowe's life upside down.
I'm bald and bruised, but I'm still fighting.
More than 2 years ago, the bone marrow disease living in her body, MDS, the same one affecting Good Morning America's Robin Roberts,
I literally fell down and couldn't get up.
turned into leukemia. Doctors told her she had a short time to live, likely months. Her condition was too grave for a bone marrow transplant. Her search for answers led to the UCSD Moores Cancer Center and a clinical trial born from a unique annual symposium, mixing academia and the pharmaceutical industry. Because of the bonds formed, researchers called up Pfizer to compare notes on a new drug aimed at other cancers.
Together, we were able to reach a conclusion very quickly that this drug may be very effective in leukemia.
Just a year later, fast for the research world, Lowe was on an experimental drug that blocked cancer cells from cloning themselves. A year later, she qualified for a bone marrow transplant. She is now in remission.
I don't know that that would have happened if this collaboration had not taken place.
So when she came to me, Linda said she wanted to see her grandchildren and spend time with them. The problem was they weren't born yet. So that was a very high bar. So she's got 3 grandkids now. She's free of her leukemia. It was a rocky road going through the bone marrow transplant, but it was an opportunity she wouldn't have had without that clinical trial. And the final 2 vignettes I'd like to talk about are newer therapies we're developing, again, as a result of CIRM funding and some concentrated philanthropy. One is the ROR1 project, where we've looked at an embryonic gene, again, that's upregulated in cancer, seems to be expressed in 94% of patients with a disease called chronic lymphocytic leukemia. And through our HALT Leukemia grant, we've developed a humanized monoclonal antibody for this. It turns out it's also very important for the spread of a number of aggressive solid tumors. So we think that the first clinical trial will be in chronic lymphocytic leukemia, and then we hope to do subsequent clinical trials with additional funding in other solid tumors. Finally, we've made a pan-BCL2 inhibitor for these leukemia stem cells that seem to survive. This is through an early translational 2 grant. So we know these leukemia stem cells are crafty, and we need to be able to get at them from different angles. So we find with this inhibitor, we're able to block survival of the leukemia stem cells. So in essence, we know that cancer is very complicated in the sense that it can evade single agents quite frequently. So I think we really owe a debt of gratitude to Bob and Alan Trounson and others in the California Institute for Regenerative Medicine for understanding that we still have a lot of work to do. And just for the last couple of minutes, I'd like to invite Teresa Blanda up to tell us what kind of work we have to do. So Teresa, you could just join us. We've got 3 minutes to go over what we've been up to. So the amazing thing about CIRM is they realize patients are part of their own solution. And rather than patronizing people, they said, okay, come to the table and be involved in the grants administration as well. And I think this has really changed how we do science. So Teresa, can you tell me about when you were first diagnosed?
My original diagnosis went back to 2003. I was diagnosed with polycythemia vera. And that kind of stabilized for, 4 or 5 years. And my oncologist at the time at Mission Viejo says, well, you know, this is morphing into something different. Your white count is starting to climb, and there's not a whole lot I can do for you now, so where would you like to go? And I said, what do you mean? So I ended up at City Hope first, and we did do a donor search and came up empty-handed. At that point, I was pretty sick, and I asked the doctor, I said, are there any clinical trials going on right now? And he says, yes, there's one in La Jolla. So I ended up going to see Dr. Jameson in La Jolla, November 2008, and I was pretty sick. I had a white count of, 98,000, and I was right on that borderline between AML and not doing so good, and Dr. Jameson wasn't sure she would take me on. And it was the last seat, too, in this particular trial, and I was like, this is my only option, what do I do? And she took me on, and as you can see, I'm a living testimonial. 5 years later, I feel pretty darn good. I'm still working. You know, I'm dating. I'm part-time caregiver for my mom, who's elderly. And I just have a different view and appreciation of life and try to explain things to people which I find they don't understand. You know, they hear stem cells, and some of the people I work with right away get crazy about, oh, you're killing babies. And I'm like, oh, God, no, we're not. You know, it's just, I go, yeah, we'll talk later. I just can't do this. But working with Dr. Jameson has been extraordinary. It's been quite a journey. And I had immediate results within, the first month. And I couldn't believe it. I had some funky tumor growing out again. I don't know what that's called.
Extramedullary hematopoiesis.
There you go.
It's always got a long name.
I had a lot of bone marrow scarring. My joints would hurt. I could barely move from the couch to the chair to the bed, and yet I was still working. I tried acupuncture, and that didn't help. And finally, through the grace of God, I was, my path crossed with Dr. Jameson, and that's been a blessing. I mean, she does extraordinary work. And I told her, whatever you need me to do, whatever you want me to do to help, I'll do it.
Thank you. So this is what I'm reminded of every Wednesday. Courageous people like Teresa, we can't let them down. And I think we're just getting started. We've got a lot more work to do. And so if we can do this on a grander scale with everybody in this room really pushing forward, I think that we can expunge some of the draconian diseases that people are having to deal with. And with Teresa leading the way, I think we'll get there.
You guys, your work is just beginning. And I look forward to continuing doing what I can as a patient advocate. I'm fully into this, and I think it's a wonderful thing. And I can't say too much about it. It's just a great thing that so many people are wanting to do this.
Thank you, Teresa.
Okay.
So you've just heard about 5 breakthroughs getting to clinical trials in different forms of leukemia from a woman physician scientist who was totally frustrated and concerned about whether she could stay in research because the NIH didn't have the money to fund her brilliant work.